Acidification and Harmful Algal Bloom webinar series. My name is Maggie Broadwater from the National Centers for Coastal Ocean Science, and I will moderate the webinar today. Jennifer Mintz from NOAA's Ocean Acidification Program will be our producer working to keep Adobe Connect going. If you have problems with Adobe Connect, please enter it into the chat box on the upper right corner of your screen, and we will try to address those. Thank you for joining us today for Acidification and Harmful Algal Blooms in the Great Lakes. This webinar series is laying the foundation for a virtual workshop that will be held August 11th through 13th, where we will discuss harmful algal bloom and ocean acidification interactions and the major questions surrounding them, and what research priorities can help address gaps in our understanding and management of these factors. This is the 6th webinar in the series to prepare for our virtual workshop. The workshop is to help us identify priorities and research products that can be incorporated into a future request for proposals on the intersections of harmful algal blooms and ocean acidification. If you are not yet signed up for that workshop but would like to participate, we're incorporating names on a wait list. Please contact Maya Sharp, whose email is in the chat box. And now Maya will introduce herself and share a little bit about her summer project with us.
Thank you, Maggie, and welcome everybody. My name is Maya Sharp. I'm a NOAA Educational Partnership Program undergraduate scholar, and I'll be completing a project based on webinar's contents, questions, and discussions. The chat box will be monitored and recorded during the webinar, and there will also be a questionnaire that we will use to inform our workshop and future directions of research funding. Those accessing the webinar online will have the opportunity to comment, ask questions through the Google form, and the link will be in the chat box for discussion at the workshop. I will compile these and synthesize them for our workshop. Thank you.
Thank you, Maya. Just wanted to take a few moments to get oriented in Adobe Connect. So audio is over the computer. You are welcome to listen through computer speakers or headphones. If you are having trouble hearing, you may either try turning up the volume on your computer or headset speaker, or turning up the volume in Adobe Connect. And you do that in the top menu bar by clicking the pull-down arrow to the right of the speaker icon and select Adjust Speaker Volume. If you are on a headset, you can go to the speaker icon, hit that pull-down, and select Speaker to find the headset. And if you are still having trouble hearing, please log off and log back on, entering using the Adobe Connect application rather than the browser. During Reagan's presentation today, you are welcome to view that in full screen, and you can do that by finding the button highlighted here in the orange box with the 4 arrows. This button toggles slide size larger or smaller. Note that you will lose chat and captioning boxes if you do go into full screen mode, but can toggle back at any time to the regular view to ask questions or add comments in the chat box. We do ask that you enter your questions into the chat box highlighted here at any time. We will hold those until the end of the presentation, and also note that, as you can see already, we will be sharing any relevant links in this box, including the questionnaire that Maya mentioned. At the end of today's presentation, we will open it up for questions and discussion. If you want to comment but are not the questioner, please raise your hand highlighted here. Click on the icon and we will unmute you so that you can join the conversation with our presenter. We will post a recording and a PDF of the slides on our workshop webpage. The address is in the chat box. And as Maya mentioned, a link to the questionnaire will be on the website for viewers to send follow-up questions and comments after the webinar. And we really encourage you to fill out that questionnaire, as we're using those responses to inform our discussion at the workshop and directions for future research. I will hand it back to you, Maggie.
Thank you, Jen. So today we are happy to welcome Dr. Reagan Errera from NOAA's Great Lakes Environmental Research Laboratory, or GLERL. Dr. Errera's research focuses on phytoplankton ecology, concentrating on harmful algal species and acidification. Her interests in HABs include identifying factors influencing toxin production, allelopathic chemicals, the impact of acidification, temperature, and impact of freshwater inflows on community structure. Dr. Errera recently joined GLERL as a research ecologist, where she is a primary investigator for GLERL's Omics program, the Environmental Sample Processor Network in the Great Lakes, and the laboratory's HAB monitoring activities in the region. During her career, she has worked with a number of harmful algal species, including Corynea brevis, Pseudonychia species, Primnesium parvum, Microcystis species, Delicospermum flossaqua at study sites spread across the world, including the Gulf of Mexico, the Western Indian Ocean, Texas reservoirs, Louisiana's Breton Sound Estuary, Barataria Bay, and Lake Pontchartrain, and now in the Great Lakes. Now Reagan will discuss acidification and harmful algal blooms in the Great Lakes. Welcome, Reagan.
Thanks. Can everyone hear me okay? Maybe.
Yes.
Okay, thank you. So thank you for that introduction. And before I start, I just wanted to thank the organizers because you've had a great line of speakers thus far, and it's just been great, and I've really learned a lot. So thank you for pulling this all together. I also want to warn everyone, you might hear thunder in the background because we're getting pounded right now by a thunderstorm, so hopefully I don't lose power and therefore internet. But if I disappear, that is why. So I apologize ahead of time. I'm knocking on some wood that does not happen. So thanks. So just to give you a brief overview of kind of what I want to introduce today is first I want to get you guys just familiar with the Great Lakes in general and the biogeochemistry within the Great Lakes, because it's a little bit different than what we've been hearing previously. And then from there kind of move into HABs within the Great Lakes, and specifically I'm going to focus on the western basin of Lake Erie, which I'm sure multiple people have heard about the cyanoHABs that we have within the region. I'm going to present some data that GLERL has been capturing over the last several years, both through our SOAR project and also our monitoring program, and then also some experiments that we have going on, and kind of see, based off of all that information, kind of where we go next potentially in the Great Lakes when it comes to acidification. And what you'll see too, basification potentially within the system. So just to get started, hopefully it goes. Maybe. There we go. So I'm sure everyone can pick out the Great Lakes on a map. It's a very distinct feature when you look at North America. These are not the lakes that are in maybe your backyard or your local reservoir or something. These are very large natural resources that we have. So just in general, we are kind of considered the third coast, where the East Coast being the most populated, followed by the West Coast, and then followed by the Great Lakes. 8 states call the Great Lakes home, and that includes 2 provinces in Canada. So this is stretching over a large range, and this also includes about 8% of the U.S. population. So lots of people call this area home, and I have to laugh slightly because I come from the Gulf of Mexico, and they used to claim that they were the third coast, but since I've moved up here, I've kind of realized that, well, maybe the Great Lakes really are. So it being situated, or the Great Lakes being situated between the U.S. and Canada, they've come up with the Great Lakes Water Quality Agreement, where both countries work together in order to manage this resource. And I'll touch a little bit more on that and a little bit later in my talk. But you can see here we have 5 basins that are considered the main portion of the Great Lakes, and they're currently under their current water levels. They are all interconnected, and this is important because that also helps determine the biogeochemistry that is going on between the system. So like I mentioned before, these are large natural resource structures that are about 20% of the world's freshwater supply when you think about that. And if that's kind of hard to kind of visualize as opposed to like the Atlantic Ocean, the water in the Great Lakes can cover the continental United States in about 10 feet of water. So if you think about how much water that is, this is a huge amount that we have. So in a lot of ways, that then determines how they're functioning, and they function a lot like ocean basins because of that amount of aquatic habitat that they have. The Great Lakes were formed by glaciers about 10,000 years ago, and they're considered young when you then think about just the basins compared to the Great Lakes. So like I mentioned, they do flow from one to the other. So Lake Superior is the highest, and it flows in, and it also has a different geological structure where it's more granite-based as opposed to the other four Great Lakes, which is more carbonate-based. But Lake Superior will flows into Lake Huron, and then also joins with Lake Michigan to form Lake Huron, that then flows into Lake Erie, which is the shallowest and warmest of the Great Lakes, flowing into Lake Ontario through Niagara Falls and then out through the St. Lawrence River into the northern Atlantic. And so, like I mentioned, this kind of helps determine the biogeochemistry within the system, and I'll talk a little bit more about that as I go along. But we are, the Great Lakes experience different changes in water level, just like we are seeing with coastal systems in the oceans. But a little bit of a difference here is that the water levels here are not changing as much with thermoregulation or thermal expansion as like we're currently seeing in the oceans, but more from increased runoff. So we've just finished a period between 2001 to 2013 where we were considered at record lows within the Great Lakes of water level. And we've now entered since 2014 to a period of record highs. In fact, we've had all 5 lakes have had record-breaking highs in water level over the last several years. And this, like I mentioned, it is mainly impacted by the precipitation. So we've seen increases in mean precipitation within their drainage basins, which then affect this level of water within the basins themselves. So as the water level goes up, that's going to change the chemistry slightly of the system, but also that really connects the basin, the Great Lakes Basin to the water shed that they are associated with. So a lot of that's coming down within the system. So we've seen an increase in precipitation, and I'm just pointing out here, I think you guys can see my cursor. Now you can see my cursor. Maybe, hopefully. I'm just pointing out here with these circles, 2 basins that we know are really directly impacted, both Saginaw Bay and Lake Huron, and then the western basin of Lake Erie, that are really connected to that terrestrial input that is associated with that precipitation. We also know that temperatures are rising within the Great Lakes region, and that Lake Superior is seeing the largest increase in temperature, and Lake Erie having the smallest increase. However, it is also, like I mentioned earlier, the warmest of the Great Lakes. So to look at the biogeochemistry a little bit of the system as a whole, Phillips et al. Back in 2015 did a review. And there has been a lot done even since this review of where we are as far as acidification is occurring in the Great Lakes. So as I mentioned before, the basin itself kind of determines some of the biogeochemistry in that Superior with that granite base is considered more acidic, but it also has a lower alkalinity as shown by the table than the other calcium-based type basins, or the 4 other basins. Regardless of all that, the basins actually act very similar to the oceans, and we can see this in 2 main areas. First, primary production, in that oligotrophic systems such as Lake Michigan have a very similar primary production than we see in places like the Aloha Station off of Hawaii. And Lake Erie, which is more eutrophic, is very similar in primary production to our coastal upwelling areas, or large areas with freshwater inputs, such as the northern Gulf of Mexico. So these are reacting similar to what we see in the oceanic basins, but also just the biogeochemistry impact. So along the coasts of the Great Lakes, they are affected by coastal processes, but then once you get into the kind of meat of the middle of the Great Lakes, the chemistry is much more controlled by atmospheric conditions, evaporation, things that are going on there, so it's much more similar to the oceans. So one of the things that the Phillips paper kind of did was they looked at modeling and noticed that, yes, if we put conditions for future climate scenarios, the basin should become more acidic, but when you warm it, it actually is going to become more basic. But they also noted that there seems to be, based off the data, that there's huge variation in this over time. And part of that is that the only real data that they could look at over a large span or large portion of time was that with the EPA biannual sampling that they were doing, and they're just looking mainly at pH and alkalinity, and they couldn't resolve the differences between what they were seeing in the model and what they were actually measuring. So they kind of concluded that there really needs to be more data collection and the correct data collection out there to kind of get at what is going on with acidification. You know, that classic statement of all of us researchers, right? We want more data, and so on and so forth. So looking a little bit more at specific Great Lakes basins, Superior we by far know the most about when it comes to what is going on with the carbonate chemistry within the system. And Miner et al. In 2019 came out with a paper where they looked at the basin over 20 years and also over a very specific 3-year period where they looked at pH, temperature, and chlorophyll A. And they basically determined that the pH wasn't changing within the basin, and that was because it was being driven specifically by the increase in the alkalinity that has been occurring in the basin over the last 20 years, but also due to primary production and the water column warming. And that they did see seasonal shifts within the pH, but the seasonal shifts, so like in the spring they had a lower pH that was then a CO2 source. The basin then moved into the summer where it warmed up and was pretty much at equilibrium with the atmosphere, and then the fall went back to a slight source of CO2. And this variation was very much tied to the terrestrial inputs and the biology that was going on in the system. So I went the wrong way. I apologize. Sorry. I was like, I should be on Lake Michigan. So then when we look at Lake Michigan, Harvey Bootsma out of the University of Wisconsin, Milwaukee, has been able to put on a CO2 flux system on the Lake Express. And what the Lake Express is, if you guys haven't been to the Great Lakes, is it transverses between Muskegon and Milwaukee 4 times a day during ice-free periods. And this just, allows people to transverse instead of going through Chicago across the lakes a little bit easier. And with this flux system, he's been able to get large amounts of data to see how the CO2 changes in the system. And one thing that he's noticed, and I think my pointer is now back up and hopefully showing, is that there's large variations per day in the system, and that some of this might be tied to the temperature. But also, he's seeing fluctuations over the course of the season, right? So early in the season, early in spring, we have high PCO2, which would be then low acidity in the system, moving into a more basic system as we go through the summer. And this is really key because this kind of leads to potentially carbon limitation within the system itself, not only just being potentially impacted by the biology, but the biology then being limited itself. And one of the things that he's pointed out is that we really don't have enough data, again, to say what's going on during the periods that we're not out there, so more ice periods, so starting in November going through to that May period. And he's looked at that through municipality intakes, and what he saw was a huge increase in the PCO2 within the water column during kind of this fall turnover before the ice kind of comes over. And this really needs to be looked at more and further within the basin itself. All right, so before I move on to HABs, it would be a detriment if I didn't also mention other biological impacts in the system, and mainly the invasive Dreissena mussels. So the mussels themselves are calcium carbonate based, so they're going to be impacted by the acidification or basification that is happening in the Great Lakes, but they're also going to be impacting that basal food web that we're looking at. So the lower portion of the food web where they're feeding. And we've seen this in multiple ways, mainly in places where there is large populations or concentration of mussels. They have actually changed the community structure. And Beth kind of talked about this a little bit in her talk on Wednesday, in that acidification is going to kind of shift the community to smaller phytoplankton. And we're seeing this in Lake Michigan, where the mussels, though, have changed the community to be more pico or nano-based over time versus maybe the acidification having that control. And one of the interesting things, too, with the mussels is that they, like I said, are calcium carbonate based, so they can't, are having issues, which might be a good thing, invading into certain areas, such as Lake Superior, which is more acidic because of the low aragonite saturation state within the system, as well as northern Lake Huron, because, again, that's being fed by the water coming in from Lake Superior. So that's kind of limiting their range in some ways, but if basification could occur, they could kind of come in and tackle those new systems. In addition, we also know that these mussels are selectively feeding. And this has led to some of the nuisance blooms we've seen, not so much selectively feeding, but clearing out of the water column. But they've also have shown that they selectively feed against Microcystis, and that they're not taking them up. And can actually, there's video of them spitting Microcystis, the major hab, out. So it makes an interesting dynamic. So moving on to HABs a little bit, when we think about the major difference between the Great Lakes and the oceanic systems that we've talked about in this series with HABs, is that we drink our water. So when a HAB occurs within our system, it's not just the recreational inputs or the impact to feel like, or of the fisheries within the system being impacted by that HAB. It's also a whole different level of human health that we have to consider, because we do actively drink this water and use it as a major natural resource within the area. So just some quick kind of viewpoints, if you guys aren't aware of what kind of blooms we do suffer from. They are mainly cyanobacteria-based within the Great Lakes, and Microcystis is probably the most well-known or most recognized. But we also have different blooms in different areas of the Great Lakes, such as Lake Superior as more an Anabaena-based bloom. And even in lakes where it's mainly Microcystis, such as the Western Basin, we get large Plankthorax blooms within Sandusky Bay, for instance. So it's not this one bloom type that we're experiencing within the system. On top of that, like I mentioned before, we also get nuisance blooms that are coming into the area that not only are not necessarily making toxin, but are considered muck within the system. But like I mentioned earlier, I'm going to focus specifically on the Western Basin of Lake Erie and those Microcystis blooms that I think a lot of us are familiar with. Also, a lot of people have talked about in other talks of like, oh, Microcystis, we'll get to that later. Well, I think we're finally getting here in some ways. So when we think about the Western Basin of Lake Erie, it's mainly impacted by three major tributary sources. And the system as a whole is really impacted by the water coming in from the terrestrial system, so these water basins, or these watersheds. And one of those watersheds in particular is Lake St. Clair, that comes down through, I don't know if my arrow is actually working, so hopefully it is, but basically coming down and through the Detroit River and causing what we see is the Detroit River plume in the northern area of the basin. And that, again, that's water coming in from Lake Huron, just to connect it all through the different basins. The other, another major water source in the Western Basin is the Maumee River, which you guys may have heard about before. And this is a major source kind of coming into the southern portion of the basin. And just to give you an idea of the land use within this area, it's about 65% of this area is cultivated crops. So we're talking agricultural lands within here. And then also we have the city of Toledo associated within this basin. And then we also have the Sandusky River coming in a little further east, and that's going directly into the Sandusky Bay and then out into the greater portion of Lake Erie. And these kind of three really contribute to what we are seeing in the chemistry. And you'll see that in a little bit later when I show the data of how we're seeing acidification and basification happening in this area. So I like to show this diagram because it really indicates what is happening when these water flows come in. And this is just a MODIS image from this year. So after a major flood or rain event and flooding event within Michigan and northern Ohio, where several days later you can see these water sources kind of coming in and all meeting within the Western Basin itself. And so again, coming out of Lake St. Clair, developing that Detroit River plume, you can kind of see it located here where this is really low nutrient water by the time it gets to the Western Basin. And then the Maumee coming in with these sediments. With those sediments also comes typically high nutrients coming and playing into the system, and then Sandusky Bay down here also you can see a large amounts of sediments. But this also kind of shows how then they move around the basin as a whole. And that this is mainly driven, this is a shallow basin, so mainly driven by wind events pushing the water around within the system itself. So we've had an algal problem within this area for a while. In the 1960s and 70s there were algal blooms, but then we had phosphorus kind of reduced within the system up until the late 90s, or mid-90s I should say, and that's kind of indicated by the diagram over here where phosphorus was driven down and we really saw those blooms go away. But in the late 90s, early 2000s, we saw an increase in phosphorus coming into the system. And nutrients, I shouldn't just say phosphorus, but all nutrients coming into the system, and we saw the blooms come back really around 2003 as an indication of their return to the system. And because of some of this, due to that Great Lakes Water Quality Agreement, Canada and the U.S. kind of decided that they needed to reduce the loading going into the system. And Rick Stumpf and colleagues at Heidelberg have really shown, this connection between the flow that is coming out of the Maumee River and the nutrients coming into the system are really determining the size of the bloom. So a reduction was suggested of 40% of particular phosphorus and then also 40% of the DRP within the system to kind of try to get these under control. And this helps us a little bit because it also, we know from this discharge and we know from this water coming in, it's also going to affect other parts of the chemistry of the water system within the Western Basin. And so what we're seeing is that during high flow years, such as 2015 and 2019, we're getting larger blooms, and we'll look at a little bit about the chemistry here in a second and what we see there as well. But then we have moderate years, such as 2014 and 2017, which also can produce big blooms. And then smaller discharge years, which cause a little bit smaller of a bloom. Such as 2016 and 18. And this is more size of bloom, not toxicity or anything like that I'm referring to, but it can kind of give us a clue of what's going on, because there are four main things that are really impacting as far as the chemistry of the system. It's the biology and temperature here. We're also looking at those nutrients that are coming in not only from the Maumee but from the Detroit River, and then and I guess those are four right there that I just said. Sorry, I connected them all. So before I get into the data, I just want to kind of remind everyone of kind of where we are when we think about pH and acidification. And like I've been saying a couple times, basification, because this is going to play into what I'm referring to in that we're using pH as almost in some ways as a proxy for carbon and carbon limitation, because the pH and inorganic carbonate system are so closely linked. And because of that, we kind of can look at who's being stressed under these carbon limitations or carbon replete conditions. And Vesser et al. Has this great kind of diagram here that I have over to the side that shows under oligotrophic conditions, the low nutrients levels and high CO2 amounts, that the systems become nutrient limited really quick. But in the western basin of Lake Erie, we're really dealing with a eutrophic system. So instead of becoming nutrient limited, the community can become carbon limited really quick as it blooms and as it becomes the biomass accumulates. And this can also then lead to light limitation within the system. And cyanobacteria in particular have developed ways in order to deal with this carbon limitation. And that can then play into how we are seeing the bloom and how the bloom develops, which is a really interesting factor of looking at the system as a whole. So for instance, we know that some cyanobacteria species, they form scums, and one of the ideas is that they are forming these scums because they're carbon limited in the aquatic system, so in the water itself, so they pop up and are able then to take advantage of the carbon in the atmosphere. And then also they have different ways of bringing carbon in their cells through the carbon concentrating mechanism, which I'm going to talk a little bit about later and about how efficient that is. So then we can look at Lake Erie and see if the system is actually carbon stressed and how that then plays in competition with other species that or the overall assemblage within the system. And also see if Erie is a sink for carbon, which could be a good thing. So at GLERO for the last several years, since basically 2012, we've had these set stations that we go out beginning in May and monitor throughout the season up and through October. And these are 9 stations, and you can see the parameters listed for our discrete samplings once a week. And then we also have continuous stations at 4 other locations. And really, for today's talk, I'm just going to be focusing on these 3 particular stations. And the reason I'm doing that is to show how these water bodies are kind of interacting that are coming and flowing in. So station 2, so the lower station, is directly impacted by the Maumee River. It also has a bloom. You see the biomass from the bloom almost every year, or I should say every year, not almost every year. And then station 8 is along the Michigan coast, and it also is impacted by the bloom every year. But it's also impacted by this shift and mixing that is occurring back and forth in the system. It's a little bit out of the, coming straight off the Maumee, but more this mixture that's occurring. And then station 4 is actually more frequently impacted by the Detroit River bloom than the bloom itself. So not as impacted directly from the Maumee River discharge. And so what I'm going to show is a series of what I like to call graphs of graphs. But hopefully I'll point you to the major differences here. And I'm going to start with chlorophyll A and using fluorescence as a proxy for biomass from these continuous stations. And I'm showing chlorophyll A over on the left and then through time, so a time series. And then also indicating the spatial difference within the Western Basin. So 2 being the first one, 4, that one impacted by the Detroit River, and then 8 kind of being pushed around a little bit between the water mass as it shifts within the basin. And one of the clear things we see with the biomass is 2015 in particular, so that was the largest cyanobacteria bloom that we know of in the Western Basin. But at the individual stations, the biomass was rather low as compared to, say, 17 through 19. We also see that station 4 in particular has a lower biomass, and this kind of makes sense because that's being impacted by that water coming in from the Detroit River a lot more than the really nutrient-rich water coming in from Maumee River. In particular, 2017, where we had a large bloom within the basin, we don't really see it at all in this station 4 biomass. And as we continue then through, this is kind of just giving us a view of where it's blooming and how it's spatially changing through the system. We can then look at how the pH is changing. And again, kind of using pH as a proxy for carbon within the system. And one of the things that, if you noticed, we're not taking enough parameters to really get at the inorganic dissolved carbon in the system yet, but that's planned. So we're kind of using pH as proxy here. So one of the things, important things that I want to point out about what's going on with the pH is that we do see during these years, especially in Station 2, this one affected more by the Maumee River, is that it has a lower pH start spring load. So when the pH is lower coming out of the Maumee River, and we see that within the data, and especially in these very wet years where the pH is starting out more acidic. As we move through, the basic mean it is roughly the same between all stations and all years. There's not a real big difference. And one thing in particular, too, is Western Lake Erie 8 station, we did see this big basification happening in the mean, and this was during that large bloom year. So it's kind of tied in with this biology controlling part of this pH system. And another point that I just want to bring to everyone's attention is parts where the Detroit River is having an impact on the Station 4 in 2017, where we have a lower pH basically throughout the entire season in this one area. So these water systems are really affecting the pH. So it's the flow from these two water systems and the biology and temperature that is having an impact on this carbonate system. And just a quick view at the temperature, because I mentioned this, like expected, it's nice and cool in the spring, it warms up during the summer, and then it cools off during the winter, and these are from May through November. So you can kind of see this typical pattern happen every year. And when we really zoom in on this, and kind of what I've been kind of alluding to, but it's kind of hard to see when I have graphs of graphs, right? It is this combination of the pH or the carbonate system being affected by different parameters at different times within the bloom itself. So at times we have both temperature and the biology kind of driving this increase or this basification or even acidification that we're seeing within the system, say later in the season in 4. At other times, we have the temperature driving the system. So this is particularly interesting in Station 2, where the biomass is indicating that it's increasing in biomass. So you would think, okay, it's going to be photosynthesizing, it's going to be pulling that carbon out, driving the system basic. But what we actually see is that the system is actually going more acidic, and that is most likely driven more by the temperature. And then at other times, we see that the biology is really having the largest impact on the basification and acidification within the system, such as again here with Station 2, where we have large amounts of respiration most likely going on within the system because of the decrease in the biomass, driving that pH back down to become more acidic. And I just want to point out here, this circle here, the second circle in Western Lake Erie 4 should not be there, so don't look at that and be like, what is she talking about there? That was a typo on my part. So with this combination, we can then look at, how chlorophyll A and pH are kind of related to each other. And we do see a positive relationship. So as chlorophyll A goes up, mainly driven by the Microcystis bloom, we see the pH also increasing. And this is a slight positive relationship. What's interesting, though, is we also see this in the microcystin. So this is the toxin being created by the cyanobacteria in the system, which is pretty interesting. But then when you think about, and I'm just going to go back really quick, the periods of the bloom, or when the biomass is at the highest, is when we're in a basic condition. So they're driving that basification. They're showing up and making this toxin. And something else that's not captured here is that transition in the bloom that we see from toxic strains of microcystin being present earlier on in the bloom, going to more non-toxic strains of microcystis later on. And some of this interaction it is due just because of, or this particular relationship is probably due to that. So then, if we dive into this chlorophyll A a little bit further, like who are the winners and losers really here, as far as the phytoplankton groups go? So we do see this positive correlation between cyanobacteria and basification within the system, and this makes sense. Again, they're the ones kind of driving this basification. So are they making the environment better suited for themselves, and then taking away that situation for competitors such as green algae, which are negatively affected, which is the basis of this kind of diagram here that I'm showing. When we look at this a little bit more broadly, where we consider, the pH over the course of the bloom, as well as the different groups and how they're shifting, we really don't see much of a key indicator of when it goes basic, the diatoms go away and we get cyanobacteria. We mainly just see that, hey, cyanos are around when it's basic, and so are other species. So this needs to be explored a little bit further. So several of the other speakers have mentioned temperature and the clear connection between temperature and carbon and pH within systems, as well as just overall warming and how that's affecting the bloom. And Tim Davis and colleagues have done some nice work looking at this, using that 17 degrees C kind of cutoff that Rick Snupp established in his 2016 paper, of looking at how many days, or is because of basin warming, are we getting more days and a longer duration of the bloom potential during this time? And that's kind of what they were able to show through the data, is that looking at 2002 through 2017, that the bloom is initiating earlier by about 1 day, and then it can also last the duration about equivalent to 1 to 2 days longer as well. In addition, it's also the higher temperatures and the warming effect that's occurring within the basin, the growth potential for cyanobacteria is also increasing. So these kind of put together is, we can kind of think of it as potentially where we have longer time periods in which the bloom can start earlier, can go for longer, and then also have better conditions to grow under. And just then connecting, the temperature to the pH, we do see a positive relationship here. This is not surprising because of carbonate chemistry and kind of how it works, right? Less carbon can get in the system when it's warmer. So just looking at DO a little bit and some connections there. So we know and we wouldn't expect dissolved oxygen or hypoxia to be occurring in our surface waters. These are really well mixed surface waters, and the connection there with DO, because it can lead to eutrophication within the system as well as affect the pH within the system. I'm not going to go into too much detail just because of time, but we do see periodic hypoxia in our lower waters within the basin. This is our recon station just off of the Ohio coast, and what we see is that we do get short periods of hypoxia occurring during this time, and this is associated with that increase in chlorophyll A, most likely the cyanotoxins, or not cyanotoxins, sorry, cyanobacteria. But when we look at it from a threshold condition, the cyanobacteria actually do better under low DO conditions. And I know they're not really using the DO, that's not kind of the point here, but it's almost like they're making a community or an environment that is more conducive for their growth and more conducive for them to do well in. And this is just another line of evidence of that. And I'm going to shift gears slightly now to I've been talking about this carbon relationship between pH and how really when we're talking about high pH, we're talking about this carbon limitation in the system and the phytoplankton potentially being carbon limited. And we know that Microcystis in particular have ways of dealing with this through their carbon concentrating mechanism, and that there are different genotypes within the system. So like I mentioned, our strains do change. It's not one strain of Microcystis within the bloom. They are shifting and changing throughout the season. But we do know that certain genotypes of Microcystis have better way or dealing with carbon limitation, and some that are better at dealing when carbon is high in the system, so pH is low, acidic conditions. And because of that relationship, what does that mean for our strains in Lake Erie? Are they changing how they're dealing with carbon as we go through the season? And I think this is an important thing that we should look into further, of how carbon is being used. And then on top of that, how is that changing based off of the genotypes that are able to produce toxin versus other ones? And so that brings us to some acidification experiments that we're doing. So we're looking at strains specific to Lake Erie, where we're basically acclimating them to 3 different conditions of PCO2, ranging from acidified waters to basic waters, and looking to see how they change their growth rate, how they change their CCM activity, and other things such as toxin and so on and so forth. And just some preliminary results from some of this is that the strain we are using, it does have BIC-A and sub-BT, so it does better under those low CO2 conditions, and we are seeing higher activity during those low CO2 conditions that we have in the bottles. Unfortunately, right now, this work has been halted a little bit, but we're going to continue it once we get back in the lab more frequently. Another project that we have started with Tim Davis and I is we're looking across multiple cyanobacteria species. And these are important because they're making different toxins. And that relationship between toxin production and uptake of carbon. And on top of that, we're also going to be looking at temperature, because that's a clear indication of differences or that synergistic effects between the temperature and the pH range. So we're going to look again at very acidic conditions and to very basic conditions, and then how things change, such as the toxin, the growth rate, omics, so not only the species themselves, but the microbiomes changing and the natural communities. And I should say, in both these sets of experiments, we're doing these with not only cultures, but we're also doing this with natural communities, because I think that's key within our community, because we do have these shifts looking at pre-bloom, post-bloom, and bloom periods to see how they're changing. So one of the things that they asked us to do, or the organizers asked us to do, is kind of put out some grand ideas about where research could be going within these ecosystems. And my thoughts for particularly the Great Lakes is that we need, and I know I'm going to sound like the typical researcher here, saying we need more data, but it's not just we need more data. We need high-frequency data of multiple inorganic carbon parameters to really understand what's going on with the inorganic carbonate system in all of the Great Lakes. We have a good idea of what's going on in Superior, but we have 4 other basins that could be affected by increased CO2 in the atmosphere, but also increased just runoff from that precipitation coming in, and how that's then determining the inorganic carbonate chemistry within the systems themselves. And then that leads directly into these synergistic effects that we're seeing between the temperature, the nutrients, the biology that is changing it, and so on and so forth. In addition to that, we need to look at our calcium based organisms, mainly our invasive mussels. They've already changed our system, but they've changed our phytoplankton system. With acidification, could they no longer be able to thrive, and therefore will our systems rebound, or vice versa? What would happen under basification, and so on and so forth, because they have had such a large impact on our ecosystem. And then just this follow-up of these strains and looking at the natural communities and how they're dealing with carbon within the Great Lakes needs to be explored and viewed a little bit further. Not only for their competition, but how things are going to be impacted potentially in the future. So just some quick thank yous. I presented a lot of data, and I'm just one person, but this really comes from a whole group of people going out there and collecting a lot of this over time. So thank you to them, and thank you all for tuning in. Hopefully I gave you guys a good indication of what's going on in the Great Lakes, and also kind of gave you guys some maybe some triggers, some interesting triggers to think about for the Great Lakes in the future. So thanks.
Thank you, Reagan. Sorry, I was having trouble finding the mute button for a minute. I would like to remind everyone attending today to please take 2 minutes before, we're not finished yet, we do have some questions, but before you leave, please do remember to fill out the Google form that's linked in the chat box. The information that you provide in that form will, after hearing the webinar, will provide input to our discussion during the August workshop and also for Maya Sharp's internship project. We are going to open the conversation to questions now, and I think Maya is on to read those from the chat box. We can unmute you as the question is read if you would like. You can raise your hand to contribute to a back and forth discussion with Dr. Errera. And we are asking people to confine the discussion to 2 to 3 minutes per question so that we can address everything. And I think we've had a few come in already, but please add your questions to the chat box and we'll go in the order that they are submitted. So I'm going to pass it off to Maya for our first question.
Thank you, Maggie, and thank you, Reagan, for your presentation. The first question for you is from Joseph Ortiz. pH and OT are correlated due to photosynthesis, but the O2 data is also influenced by changes in temperature due to variable saturation state, which is temp dependent. My student Nick Boney studied this at Old Woman Creek. Have you considered trying to use the pH and O2 in conjunction as a productivity proxy for the system? That may be more closely related to the carbon demand.
Yes, we are. That is a great point, and maybe I probably didn't kind of point this out a lot. I should have pointed it out more, that yes, pH and O2 are definitely highly driven, especially in these large blooms, by that biology that is going on within the system. And yes, this is something that we're planning on doing, and I would love to talk to you about this more, a little bit just about this correlation. I also feel like we need to get a better handle just on the inorganic carbonate chemistry in general within the system. We're only, or have historically, I should say, taken pH, and we know pH probes aren't great. They drift. They're not the best indicator for carbon within the system, and we really you need to take 2 parameters within that system, and I think that's key as well. So yes, I totally agree, and I think we should have a conversation more. I hope that answered your question, Joseph.
Thank you. And our next question is from Gina. To clarify, in Erie, toxic strains are present earlier in the season and later are replaced by non-toxic strains. We don't have strain data for Lower Green Bay, but we see higher microcystin concentrations later in the season.
That's interesting because I did not know that. And this is too, I'm still learning the system. As I noted earlier, I come from the Gulf of Mexico, so I know that system really well. This is a new system for me. But yeah, that's kind of what we're seeing, and some of this is Tim Davis's work, and he could probably talk about this a lot better than I can. But what we're seeing is that there are more toxic strains, right, earlier in the bloom, and then it moves to these non-toxic strains coming in and replacing those toxic strains. So yeah, that would be an interesting dynamic. I know there of how that's shifting and what are the environmental conditions that are causing that shift between the 2 systems, right? So one of the things that we're doing at GLORO is comparing Lake Huron, so Saginaw Bay, to Lake Erie, but maybe we need to be pulling in Michigan as well into the system and seeing, what are these environmental conditions that are affecting all of these different blooms that are all based off the same species, right? So that would be really cool. I'm answering questions with giving everyone more questions to work on, right?
Thank you. And we have another question from Joseph Ortiz, but he also sends this thank you. He asks, I wonder if the increase in microcystin late in the season is due to cell lysis as the bloom crashes.
So yes, we do see, and I didn't show that data here, we do see an increase in our dissolved portions of the toxin later on in the season. So microcystis is, or microcystins, I should say, are very similar to brevitoxins, which make it nice for me because I'm used to dealing with them, is that they're kept intercellularly, or at least inside the colony of the microcystis. So at the end of the season, when they start to break apart and lyse, we see that uptick of the toxin, or the dissolved portion of the dissolved toxin in the water, is what I should be saying.
All right, if anyone has any further questions, please enter them into the chat. I see Beth typing, and I thought I saw Tim Davis typing, but we'll give it just a minute to see if anyone has anything further, and I'll go ahead and let everyone know that we will post a recording and a PDF of Dr. Errera's slides today on our workshop webpage, which I think Jen will post in the chat, and also a link to the Google form will be posted on that page with the recording as well. And our next webinar is going to be this coming Monday, July 13th at 2 p.m. Eastern with Melissa McCutcheon from Texas A&M University at Corpus Christi, presenting a synthesis of the existing research on ocean acidification and HABs to tell us the full consensus of what we know at this point. So I don't see any additional questions, but thank you for joining us today. Thank you, Reagan and Maya and Jen, for keeping things running behind the scenes.